Welcome to the Deep Dive. Today we're digging into a really important pan-cancer study from 2018. It looked at inherited cancer risk across thousands of patients, over 10,000 actually. And here's the main takeaway, the big number, 8%. That's the key finding. Nearly 1 in 12 adult cancer patients in this study, who weren't selected for family history or anything, carried an inherited genetic variant that likely contributed to their cancer.
Yeah, 8%. It sounds straightforward now, but it really reset the baseline.
It seems much higher than people thought, right?
Yeah.
Especially for an unselected group.
Exactly. Before this, our view of hereditary cancer was, well, pretty fragmented. We knew a lot about specific genes and specific cancers, like BRCA in breast and ovarian cancer, often from studying high-risk families.
So it was kind of skewed. How did this study get a more unbiased look then?
They tapped into the TCGA Pan-Cancer Atlas. It's this huge resource with paired tumor and normal samples from thousands of patients across 33 different cancer types.
Okay, so a massive data set.
A massive unselected data set. And their mission was basically threefold. First, pin down that true prevalence, the 8% figure. Second, see if they could find any new connections between genes and cancers.
And the third.
And this is really crucial. They wanted to test if these inherited variants actually had a functional impact inside the tumor. Did they follow the classic two-hit idea?
Right. Let's unpack that 8% average a bit more, maybe looking at their figure too. They found 853 of these pathogenic variants, correct?
That's right. And while 8% was the average, it wasn't uniform. Some cancers, like pheochromocytoma, had a much higher prevalence, like over 20%. Others were lower.
And looking across all these cancers, it once paid off. Did they find those new associations they were looking for?
They did. The pan-cancer view confirmed 21 significant gene cancer links, including some that were, well, quite novel, things that single cancer studies might have easily missed.
Can you give an example?
Sure. A key one was linking PLB2, which we knew was involved in DNA repair, to stomach adenocarcinoma. That wasn't really on the map before.
Interesting.
And another was SDHA, a gene linked to certain inherited syndromes, but they found a significant association with melanoma, skin cancer.
So finding these connections immediately changes things, right? Like who doctors might recommend for genetic testing.
Precisely. It broadens the criteria. Now let's talk about that functional consequence, the two-hit mechanism, which is often shown in Figure 3 in these kinds of papers.
Okay, the two-hit hypothesis. For tumor suppressor genes, or TSGs, the idea is you inherit one bad copy, and then the tumor needs to knock out the second healthy copy to really get going.
Exactly. The tumor has to eliminate that remaining wild-type allele.
So how did the researchers actually check if that was happening? How did they prove the tumor was actively getting rid of the good copy?
They used the tumor's own sequence data. They looked for something called loss of heterozygosity, or LOH. Think of it as the tumor deleting or silencing that protective second copy.
Did they find it?
They did. In about 38.5% of the cases involving known tumor suppressor genes, they saw clear evidence of LOH. This confirms the tumor is actively selecting against that healthy allele. You see this very strongly with BRCA1 and 2 in ovarian cancer, for instance.
Okay, that makes perfect sense. For TSGs, you need to lose the function.
Right.
But the study also found, 33 pathogenic variants in oncogenes?
Yeah.
Genes that usually drive cancer when they gain function.
Ah, yes. That leads us to a really interesting point. So here's a quick thinking question for you listening. If tumors select for loss of function in TSGs via LOH, what do you think needs to happen functionally with those inherited pathogenic variants found in oncogenes?
If the tumor isn't trying to lose the oncogene copy, maybe it's doing the opposite, trying to amplify its effect. Did they look at gene expression levels, perhaps, like in Figure 4?
You got it. They looked at gene expression, and the results were striking. Just as we discussed, the TSG variants tended to be associated with low gene expression. About half were in the bottom quarter.
Consistent with loss of function.
Right. But for the oncogenes like MET and RET variants, it was the complete opposite. They saw significantly high gene expression. Over 60% were in the top quartile.
Wow. So the tumor is effectively selecting for more activity from that already mutated activating oncogene allele. It's amplifying the gain of function.
Exactly. It's a beautiful demonstration of selective pressure working differently depending on the gene type.
The really innovative part here seems to be how they connected all the dots, doesn't it? It wasn't just finding the germline variant, it was linking it to the LOH, or the lack of it, and then to the gene expression in the tumor.
That integrated framework is the breakthrough. It takes a static finding and inherited variant and gives it functional context based on what the tumor is actually doing with it.
And this had practical implications too, right? For variants of uncertain significance, VUSs.
Absolutely. This functional framework allowed them to nominate 47 VUSs as high-priority candidates for being reclassified. Why? Because they now had functional evidence, like high expression for an oncogene VUS or LOH for a TSG VUS, suggesting it was actually pathogenic. It gives clinical labs real data to potentially upgrade those uncertain variants.
That's huge for patients stuck with an uncertain result.
Oh.
Okay, let's recap the main takeaways. First, that 8% number. It's the new baseline for pathogenic germline variants in adult cancer patients, even unselected ones.
Right. Second, the pan-cancer approach uncovered new important associations like PALB2 with stomach cancer, changing who might need screening.
And third, the tumor itself tells you about pathogenicity. For TSGs, look for LOH and low expression loss of function. For oncogenes, look for high expression gain of function.
Those are the key points.
Is there any key limitation or final thought we should leave people with?
Well, one important piece of context the authors themselves mentioned, even with over 10,000 patients, the study still had limited power to detect very rare inherited variants that might have a very strong effect.
So the true burden of hereditary cancer risk.
It's likely even higher than the 8% they could definitively confirm with this data set. There's probably still more hidden hereditary risk out there.
Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has three main goals. 1, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. 2, to build a supportive community where we can exchange the most valuable experience-based research advice. 3, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.